So unfortunately, we did not get the microphone, so I will try to speak up and hope that everyone hears me. I was really pleased to see that the top answer was that the microbiome containing samples should be stored, and this was sort of also our idea when we started the MICROBE project almost 4 years ago. MICROBE is a project, it's called Microbiome Biobanking Enabler, and it's now entering, unfortunately, in almost the last half a year, so we will be officially ending in January 2027. And some of our first outputs, there are many more to come, but some of our first outputs we already shared with you in form of these training sheets. You're really the very first audience that get them. We just finished them like 10 days ago, just especially for this event was the special push. We structure this event is, I will just give you sort of a brief pitch why we believe that the microbiome banking is missing link, also especially in the context of One Health. And then we have picked out some of the topics that we handled within the project that then Cornelia and Perrine is going to also briefly present, and then we want to really exchange with you, because MICROBE, the whole concept of MICROBE was to provide the solutions for the community and not to establish a concept of new biobanks, because we think a lot of things are out there and there are just some gaps to fill. But coming back to why microbiome biobanking and why microbiomes, it was really interesting because I think it was around 2020 when it really started this sort of recognition that the microbiome-based innovations can address a range of societal challenges. And this was not only recognized by science, it is usually done, but it was actually really recognized also by policymakers. So it was this nice sort of opinion paper brochure from FIO in 2019, from that I also borrowed these images. But then when the European Commission did the Food 2030 strategy and established the pathways to accomplish the Food 2030 strategy, the microbiome world was one of these pathways, which was really significant. And then obviously the scientific community, as I said, I have here just brought 2 examples, is really on to see that there is a potential in exploiting the microbiomes to address many things. However, and this is a lot of work that we did in a previous project, the Microbiome Support Project, that was a coordination and support action which was running between 2018 and 2022. And we really there, we did not do any research, we looked into the microbiome research landscape, the microbiome innovations landscape, the microbiome translational landscape, and tried to identify the gaps. And what we sort of did here, as I said, the data is a little bit older, but I will tell you did not change significantly, is that we saw an explosion of the microbiome-related research in the scientific publication, and we did not only here query the word microbiome, but also microbial communities and all of the synonyms that were used before microbiome became a term. So you really see this explosion, and we all know that this explosion came along with the sort of high throughput sequencing methodologies. But what we then did, on the other hand, is to investigate how correlated the fields are. So we screened all of these publications and sort of investigated what's their shared base of knowledge based on the references that they cite, and we saw there's sort of none. We really saw that these fields are really siloed. So the soil microbiome community will sort of build their methodologies, their knowledge, the references on other soil publications, but they will not have a lot of sort of contact point with anything in human or even in other environments. And why is this influence on its quality, and ultimately it will end up in human health? So this is why it's really important to make this microbiome science much more unified, to also learn to speak the same language and to work much more together than we are doing nowadays. Also, and this has been again known since years, is that all of the higher organisms are essentially holobionts, so none of us would exist with our own microbiome. Most people know, I mean, most of you are from the human field, but also every plant, every animal, everything has its own microbiome. And without that, also the plants will have problems to grow and prosper and be nutritious. And this is also why we need to also not only then study microbiomes, but also focus on their interactions with the environment and the holobiome they are in. Another gaps that we have established is, and this is also a little bit probably connected with the funding, which was also one of the points you had, is what do we know and how do we study microbiomes. And I have been updated this right image since the start of the project, and these are numbers I just took off the Magnify website 2 days ago, but they never, I mean, the numbers grow, but the relationships don't change. So what we see is obviously that the marker genes, the ribosomal RNA or the ITS for fungi, we have a humongous number of those data sets, but they just give us a sort of a snapshot of taxonomy. If we want to go into functionality, we need a metagenome data, we need a whole genome data, and we have only approximately a tenth of those available in the public data sets. So that's why we are still really at the sort of correlation-based and not really in the causality-based, because with the high throughput sequencing, we are also with the 16S rRNA, we are sequencing a part of what was once a marker, so we are sequencing a marker of the marker, and we all know how, or most of us know how limited the data that's coming is. The second problem that we still have, and there were some questions and you answered that one correctly, is we still cannot cultivate most of the microorganisms that are out there. Yeah? And this is also why it's important to think about how to preserve samples where they live in, because we obviously cannot cultivate and cannot preserve them on their own. And I'm really happy, and I will also highlight this later, but until last year, the microbiome was never on this biodiversity debate agenda. We were always talking about butterflies and panda bears and whatever, and they're important, but every time thinking back to holobiome, we lose a higher organism, we lose its whole microbiome. And now it's really important that finally also the microbiomes or microorganisms came to this agenda. But again, if we cannot culture them, how can we preserve them? Then we are back to preserving the microbiome containing samples, so that's why it's also important. This is from which one of our colleagues, Matt Ryan from Kavi, who is also involved in the project, calls state of the art. This is the sort of the biobanking and the culture collection landscape as we had it at the beginning of the project, and this is that we have a culture collections that will preserve living organisms, sort of like an exenic cultures, and they're rarely stored as a consortia or more complex samples. Why? Because we don't know how to preserve them. It's an easy answer. And then we also know that, and we have already seen it in the sequencing data, that some biomes are more sort of prioritized. There's some other biomes. So we also have here a little bit different image. And then we have sort of what we sort of call biobanks in the project that collect like materials. But then this, when you collect, I don't know, when we just think about the seeds, they think about the biobanks preserving seeds. I'm now thinking Svalbard. They think how to preserve the viability of the seed, but they're not thinking about the microbiome in the seed. So are we doing the right thing in the context of holobiome? And then the last biggest gap is really linked between the biological samples and the research data. We all swear to the fair data in each of our proposals. The problem is the data is not really fair. We have a lot of sequencing data, but then if you try to dig deeper and search for the metadata, you will often find only really basic information, which makes reusing and reproducing these studies really difficult. And then, just also collecting some connecting biological samples with some downstream or upstream studies gets more and more difficult. So these are the gaps that we have, and without addressing these gaps, we will not be able to exploit the potential we have in the microbiome. You can think about many other reasons why we would need a microbiome biobanks. We can provide resources to underpin research. We can make sure that we have deposits that ensure compliance with legislation. We can use it for the protection of IP or the biodiversity conservation. There are many reasons, but we also need to know how to do it. And this was the example I brought before, that we now finally have the global microbial biobanking, sort of microbial conservation specialist group that advocates for the microbiome preservation. But for example, we also had since 2025, end of 2025, the soil monitoring and resilience law that actually prescribes quite an extensive sampling and regular sampling of European soils. But what do we do with them? How do we preserve these soils properly to preserve their biodiversity? This is what our ambition was 3.5 years ago, or even 4 years ago, but we wrote a proposal. And it was on the one hand really focusing on the technical gap, so really looking about how to preserve this diversity, both looking at taxonomic but also at the functional level and not looking only in the bacteria. Also, what would be the optimal collection and the preservation protocols? How could we do the targeted isolations? We cannot isolate everything. We all work on the limited resources. And then, but we also looked, and you will hear part of that story today because we thought perhaps this is also really important for this audience, is what are like these sort of other requirements, talking about standardization, talking about legal and ethical guidelines, talking about the business opportunities. If you have an isolate you can multiply and always resell, you have a really sort of clear business model. If you have something that you cannot really re-amplify, what is your business model as a biobank of a culture collection? So those were the issues that we also tried to tackle, and not on our own, but always in interaction with the relevant stakeholders. We asked you some of these questions, you gave us your answers, and I think we can make now an argument for any of these points, but I think it's not only important to think what should be preserved, but what is the purpose of preservation? Because if I really want to have a snapshot in time, I will treat sample differently than if I want to keep it for a subsequent cultivation in 3 months. And then a bunch of other questions that you might ask yourself is, of course, not only the question of how to access what you want to access or how to preserve it, but also how to access the compositional and functional integrity of the complex sample, or how to link this metadata with the biological samples and keep the provenance. But also, if you have a limited sample that you cannot amplify, who should have an access? How do you share the samples? Who decides? We did it in a sort of, we addressed this from many different sides, and we were really happy in the project that not only we had the partners, but we had a strong link with the different research infrastructure. So we had links with the BBMRI through our colleagues at the Medical University of Graz, but we also had links with MIRI through INRAE and also to EMBRIC through the colleagues from Sorbonne that also joined us here today in the audience. Also the organizations like DSMZ and CABI that have their own collections were on board. So we could really exchange. So this was not just a project made by scientists in an area we don't understand a lot, but really always trying to go in a closed and early exchange with the relevant stakeholders. Because our idea was never, as I said at the beginning, to build up something completely new, but rather to see how the existing resources could be used and connected in a better way to provide, benefit for everybody involved. This is a quick overview. We are going to go together today in 2 deep dives. One that will focus more on the standardization issues, because we thought that it would be really interesting for this audience, and I think you will. It might be interesting to hear what Connie has to say, and then we also will present not so much about the microbiomes, because they're so perhaps a little bit even further out of reach, but really focusing on defined microbial communities as a sort of intermediate step between the single isolates and the complex microbiomes, and also thinking a little bit, and we will present you what we elaborated with other expert group, and we would love to hear your feedback and opinion about the business models about that, and how could it look for the biobanks and the culture collections. With that, I want to thank you for listening to me. We will take all questions after the 3 pitches, so just keep posting them in the Menti. And obviously the great team behind this project, because without them most of the work would, or all of the work would not be possible.